So here's the more information about homologs, orthologs, and paralogs. So the homologs have a common ancestor. So we believe that there was some living organism, and then that living organism underwent mutations. And so in the end, everything that exists goes back to that first organism, right, which was much simpler, but it evolved over time. And because they have there is sequence conservation, then we think that there was an original sequence that just changed over time, but it came from something originally the same, and that's when we say that the sequences are homologs. You might learn in other classes about convergent evolution, which is like, okay, the birds have wings, so all the wings are homologs, but then the wings of bats are different. So the DNA sequence for a wing of a bat is not going to be similar to the DNA sequence of a wing of a bird, because the wings of bats are more similar to arms. They're mammals. So in that case, even though they do the same function, they won't have the same sequence. They're not homologs. When we talk about homologs, it's more like there was one DNA sequence that they had in common. All right, and then here are examples of orthologs. So remember, I told you that orthologs. So orthologous means like a cross. So you could think that orthologous is a DNA sequence that happens across different species. Okay, so it happens in frog, chicken, mouse. Paralogs, think parallel, right? So they occur in parallel within the same organism. So when I say mouse alpha and mouse beta, I don't mean two different mice. I mean that the same one mouse, that same one animal, has two different sequences for this globin gene. So there is a globin alpha and a globin beta. How does that come about? So there is some gene which duplicates. So there's a gene duplication event, and then let's call them the alpha globin and the beta globin. And that alpha globin exists in frogs, chicks, and mice. And the beta globin in frogs, chicks, and mice. So all of them have both versions of globin. So if you compare the frog alpha globin with the chick alpha globin, you're comparing two orthologs. And if you compare the mouse alpha globin with the mouse beta globin, you're comparing two paralogs. If you compare the mouse alpha with the chick beta globin, you're making a mistake. So that's not how, or you are going to compare and they're not going to actually group together. So let's see. Normally, when we look at orthologs, the sequence that does something here, so this globin alpha does something very similar to chick alpha, and the paralogs, they don't necessarily have the same function. So the mouse alpha will do something similar but not quite the same as the mouse beta. So it could be it's a globin that is expressed, it's a gene that is expressed as at one developmental phase of the organism, and the other one is expressed at another developmental phase. They may have similar roles, but not be expressed at the same time in the development, or they actually don't, even their function could have diverged. So when people are doing research, they're like, have you found a homolog? Which is basically saying, have you found other organisms that have the sequence that you are studying? Because people have their pet DNA sequence. Or they are asking, in that one organism, have you found another version of it? So when they ask, have you found homologs, it's mostly a way of asking both of these questions, orthologs and paralogs. So good. In homework, you're going to have opportunity to practice this thinking about speciation and gene duplication. Okay, so I mentioned the consensus sequence. I'm going to show you an example. Here is a bunch of, I don't even remember what this was, virus or whatever it was, but a bunch of places, I think in China. And let me explain to you how you read an alignment. So this is a multiple sequence alignment. And okay, this first row here is the nucleotide sequence that came from that sample of Hebei1. And then this other row here is nucleotide sequence that came from that sample from whatever that place is. So when we do a multiple sequence alignment, you're comparing all of these different samples. And then in the last row here, you have what's called the consensus sequence. So almost everything was aspartic acid. So this is aspartic acid. And then this one is asparagine. It's similar, but not exactly the same amino acid. So then in here, there is some symbol that is to convey the idea that it's different amino acids, but very similar. And then the second amino acid is a bunch of S's. So it's all serine. They're like, oh, everybody likes pepperoni or whatever. So then there you go. We are going to order pepperoni. And then the other one is phenylalanine. Everybody likes mushroom. So there's then that. So the consensus sequence is when you compare all of these, what are the positions where the nucleotides agree? And then where are the positions where they diverge? So this is like one example I wanted to show you. And well, like in the homework, you will have an opportunity, which is if I'm looking for, let's say I'm looking for, oh, my cousin was asking me this. She's doing COVID tracing in California. And she was asking me, how do people figure out if they have a different strain? And then I told her, you could do PCR that is specific for one strain, or PCR that picks up any strain. So when you do PCR is a lab technique that if you're going to pick up any strain, you would design primers for the parts that they all agree. And then if you want to do something that is specific, then you would want to do primers where they differ and you're looking for a specific one. So more on this on homework. So let's see. And here I'm starting to tell you the idea that there are amino acid substitutions. So here a serine is substituting for an asparagine. So wherever the amino acid doesn't agree, then we call that a substitution. And that becomes important when we are comparing proteins, because you're going to do this alignment by hand, not multiple sequence, but you're going to do just two of them. Let's see. Maybe I'll go like 2 more slides and then show you the exercise where we're going to start on Monday. So in the amino acid substitutions, they can have no effect or little effect, or they can completely make something not work or work differently. You could think about that as like sickle cell anemia. It's a one amino acid change, and it changes the shape of the red blood cells, and it changes how much oxygen it can hold, and it also can change how much malaria it can hold. So when we substitute amino acids, the protein can do something different or just break and not to do whatever it's designed to do. We use something called substitution matrix to look at the probability of one amino acid changing for another. The amino acids that are similar, they substitute for each other frequently. And I'm going to just put this up here, and that's where we're going to pick it up. So we use a matrix called BLOSSOM. It's blocks substitution matrix. I love this thing for many reasons. One of them is that it's husband and wife that published that. That's super cool. That's like dream come true for me. And what they did was they compared 2,000 blocks of aligned protein, and then they saw how often does one amino acid substitute for the other, and then they came up with scores. So we're going to pick up on this on Monday. And that's going to be this exercise that I was going to make you do. Maybe that's what I'll do. Yeah, you can start it, and then on Monday I'll ask you, what did you come up with? So this is what I'm talking about. Yeah, wait, back. This one, I want you to compare the methods of amino acid grouping and then see what you find in common and what is different. But I'll go back to here, but you could stare this a little bit and see if you find any patterns.